Hello everyone and welcome to our first abberior Instruments Summer Symposium. My name is Janina Hanne and I'm the Scientific Relations Manager at abberior Instruments, and I will guide you through today's program. The abberior Instruments Summer Symposia is a webinar series each Tuesday in July with different topics on super-resolution microscopy, and I'm extremely happy now to welcome you to the first event. The following webinars will cover adaptive optics STEP microscopy, STEP paint, and life-size STEP microscopy. Each Tuesday, we have 1 to 2 excellent speakers, and afterwards, we show you live on our microscopes how we have implemented what you actually heard in the talks. No worries, if you haven't registered yet for the next event, we'll send you the registration links tomorrow in the follow-up email. But before we start, here are a few official words from our CEO, Gerald Donnert.
I'm very happy that you joined today our abberior Summer Symposia. My name is Gerald Donnert and I'm the CEO of abberior and abberior Instruments. There's one important thing about us. We are 100% owned and managed by scientists. Among them are the following, and all have a PhD in super resolution. Benjamin Hake, more than 15 years of super resolution experience and more than 10 papers. Christian Wurm, more than 15 years and many papers. Andreas Schönle, more than 20 years of super resolution experience and more than 30 papers. Lars Kastrup, more than 15 years and more than 20 papers. Matthias Reuss, more than 15 years of super resolution experience and many papers. Joachim Fischer, more than 10 years and many papers. Tim Groth-Johann, more than 15 years and many papers. Myself, more than 15 years and many papers. Alexander Egner, a shareholder and advisor, more than 20 years and more than 50 papers. And, of course, a shareholder and advisor, Stefan Hell. Our joint passion is super resolution. To be honest, we cannot do anything else but microscopy. We live microscopy and we love microscopy. And now have a good time with our symposia. Stay healthy and see you all soon again in person.
Thanks a lot, Gerald, for the kind words. Today's seminar is about MINFLUX, super resolution post-Nobel. And we have 2 brilliant speakers who introduce us to the world of MINFLUX imaging. So our program looks as such. First, Nobel laureate Stefan Hell will talk about MINFLUX and show some of the latest results from his lab. Afterwards, Christian Wurm, our head of applications at abberior Instruments, will show you live MINFLUX imaging on our abberior Instruments MINFLUX microscope. Please don't hesitate to ask any questions in the questions tab of this webinar software. We'll answer as many questions as possible after each of the presentation. And if this is not possible, we will follow up via email and answer your question. So now we are extremely happy to have Stefan Hell here. Stefan is now director of 2 Max Planck Institutes and the pioneer of super resolution microscopy. He won the 2014 Nobel Prize for the development of super-resolution STEM microscopy, and since then he didn't stop pushing the method even further. With MINFLUX now, fluorescent imaging at the size of a molecule is possible. But see for yourself. Stefan, I'm really looking forward to your talk. The stage is yours.
Thank you very much, Janina, for hosting me here. It's a great pleasure to be here to give this webinar about something that is very close to our heart, as a matter of fact, namely MINFLUX. It's one of the top research fields in my lab, and it's a new paradigm in nanoscopy and of tracking of single molecules. Now, why is it a new paradigm? Because the way it localizes single molecules is very unique. It's quite different from the way it is usually done. And in this webinar, I'm going to explain to you the basic rationale of MINFLUX localization and of MINFLUX nanoscopy. If you combine, of course, localization with single molecule switching, then you get the images with the highest possible spatial resolution. Now, of course, I'm making a disclaimer because I'm linked with abberior and abberior Instruments, and I have to disclose this here because of best practice reasons. I'm starting out now by, explaining to you the things that you know already, namely the way molecules are usually localized. Now, the usual way of localization, this is what people usually think of when they speak about localization, is the following. You have a molecule, which is shown here as a little star, and then usually you have an epifluorescence recording, you have an epifluorescence setting, you have, say, a wide field illumination, and then this wide field illumination, of course, is projected into the sample with this single molecule. Could be a single activated molecule, could be just a single molecule per se, of course. And then the single molecule produces a spot of diffraction, of diffracted light on a camera, and the camera consists of many pixels. And what will happen is that the camera will, of course, record the diffraction blob. Some people call it point spread function on the camera. And then, of course, the position of the molecule is found out by calculating the centroid of this diffraction blob. That's what people call localization. Now, I'm sure every one of you knows that the precision with which we can locate the molecule scales inversely with the square root of the number of fluorescent photons that are detected by the camera. And so this is a usual way of localizing molecules. And now you can imagine that this usual way of localizing molecules is, as a matter of fact, not efficient in terms of fluorescent photons. And this is a huge problem because, of course, many molecules bleach. Of course, you're limited in localization speed if you do it in that way. And there's other problems coming up, like issues that are related with the orientation of the molecules. And this is why we had to think about a totally new way of localizing individual molecules. And this is what MINFLUX is all about. Now, contrary to, say, normal localization, we do not have an epifluorescence setting in this case. We take advantage of the fact that we can structure, so to speak, the illumination beam, the excitation beam, and using such a structure, and the simplest structure would be a donut. Of course, we can define a coordinate in sample space. And this is actually what is shown here. Now, in a typical MINFLUX setting, we have an excitation beam that is shaped as a donut. So it's not like instead where the de-excitation beam is shaped like a donut. So it's the excitation beam that is shaped like a donut. And the green, say, donut now consists of photons that are capable of exciting molecules. Now, such a donut, of course, very effectively determines a position in a sample. It defines a position in the sample very, very effectively because it consists of many excitation photons, and we can define a zero here, a zero of excitation intensity right here in the center of the donut very, very effectively. And not only that, not only we can define this coordinate here very, very effectively, we can also find out if a molecule is coinciding in space with this central position of the donut, with the donut zero, because if this molecule is slightly off from the zero of the excitation beam, the molecule will start emitting fluorescent photons. But if they coincide, of course, in space, there will be no emission because at the central at the center of the donut, where the intensity of the donut is zero, there won't be any excitation because there is no excitation photon arriving at this point. And so we have a very, very effective way of predefining a position in space using a donut. And remember, in the epifluorescence setting, that's not the case. We have uniform illumination. There's no way of defining any position in space. Now, the donut is also very effective, as I said, in finding out where the molecule is located. And I'm trying to motivate this to you by giving you a little thought experiment, a Gedanken experiment, as it's called in German. And so I'm introducing here a little demon. And this little demon, of course, knows something that we humans cannot know. But if we do this little thought experiment with the demon, of course, we learn something that, say, was not realized before the time this concept was introduced. Now let's imagine we have a little demon that acts on the beam deflector that is capable of shifting around the donut in space. So remember that we can shift the donut in space around because it's shifted using a beam actuator. It could be a galvo mirror like in a confocal microscope or electro-optic, say, beam deflector. And now let's assume the molecule starts moving. So what would happen? If the molecule starts moving, of course, we could in principle, and this is what the D1 actually can do, match the position of the molecule with the center position of the donut. So this little demon knows, kind of, because it's very savvy, of course, and very clever, the demon knows where the molecule goes to, and then, of course, it can match the position of the molecule with the center position of the zero. Now, why is this an interesting experiment? Because it tells you that at least in this limiting case where we have a demon, of course, you can imagine tracking the position of the molecule with ultimate precision, even without requiring the emission of a single fluorescent photon. Because in this case, of course, we can just use the coordinates that are basically, used to shift the donut around as the position of the molecule, because the coordinates that are, so to speak, imprinted on the position of the donut must be identical with the actual coordinates of the movement of the molecule. And so the demon can actually trace the molecule without requiring the emission of a single fluorescent photon. And that's the conceptually highly intriguing insight. It means that in this very limiting case where we have a demon, we can precisely localize the molecule in space without requiring a single fluorescence emission. And just contrast this with, say, the normal way of localizing molecule, where you need many, many photon emissions, in fact detections, in order to localize the molecule precisely. But here, you can have basically infinite precision, so very, very high precision, without requiring any fluorescence emission. And there's another interesting aspect that comes in, if you think about it. At the end of the day, what you do here is you match, if you will, 2 points. The molecule is sort of like a point, and of course, the zero, or the donut center, is also like a point. And if you match 2 points, you can imagine that in first approximation, the wavelengths of the excitation beam, or the diffraction size blob of the excitation beam, doesn't matter. So the wavelengths in the end will not play a major role. In fact, in the limiting case, no role at all when it comes to localizing the molecule. And this is also not a case, say, in a normal localization setting, where, of course, the wavelengths of emission scales linearly with the precision of the localization. So these are 2 fundamental, say, shifts in the way we think about localization. We get rid of the requirement of having many fluorescence detection, and also we get rid of the wavelength dependence. So this is quite revolutionary when it comes to localizing molecules. Now, in reality, there is no such thing as a demon, of course, and but on the other hand, we can think about how we can sort of mimic the situation of the demon, how we can approach this situation, this ideal situation of having a demon with reality. This can be done, of course, by implementing a proper electronics, say an electronic controller. This is shown in here. And this controller detects, of course, the number of fluorescence emission that result actually from the molecule. If the molecule is slightly displaced from the center of the donut, or I should say if the donut center is slightly displaced from the position of the molecule, there will be, of course, a fluorescence emission. And this fluorescence emission will be indicative of the position of the molecule, because if the molecule is far away from the zero, then there will be many fluorescence emissions. If it's just at the center, then there will be no fluorescence emission. And so this information can be fed into the beam deflector, into a galvo or electro-optic deflector. And then, of course, we can try to shift, of course, the donut around such that the zero of the donut comes close to the molecule. So in essence, we can kind of approach the demon-like situation by, say, trying to match the position of the zero with that of the molecule, of course. But this is, again, much more effective in terms of using the fluorescent photons, simply because the fluorescent photons here are used only for refining the position of the molecule. We don't have to use the fluorescent photons to start the localization from scratch, as it is done on a camera. We just need a few fluorescence photons to find out how far we are off from the position of the molecule. So how far the center of the donut is off from the position of the molecule. And this is why, say, localizing with a donut that is positioned, so to speak, in sample space is very, very effective in terms of using the fluorescent photons. And so we get much higher precision. Of course, if you localize a molecule with a donut, even though we may use, say, much fewer fluorescent photons. Again, because we need the photons only for finding out the distance actually from the zero of the donut with respect to the position of the molecule. And that is, of course, much, much smaller than the big point spread function that would be produced on a camera. So again, we don't use a camera for detection. We have, say, a confocalized detector, if you will, and we use those photons just for the refinement. Another way of thinking about it is that the burden of localization, the burden of requiring many photons for localization is now not on the many fluorescence emissions. So it's not on the dye that needs to be bright. So this burden of requiring many photons is actually pushed on the excitation beam. But the excitation beam, of course, has zillions of photons, so there's no issue. So you always require many photons to localize something. But again, here you need those photons on the side of the excitation beam. Nonetheless, the excitation beam is not very bright. You shouldn't think about a strong intensity donut. This is just a normal excitation beam, just the way you would have it, say, in a confocal microscope, because you just need to produce a few fluorescent photons. Okay, now how does this translate into a real instrument? You've seen that we have a controller, of course, we have a detector, we have a beam deflector that is kind of, steered by the controller. Now, in the end, you have to have an algorithm that finds out where the molecule is actually located. And I'm trying to explain this to you in one dimension, because by explaining it in a single dimension, you will already understand the basics of the MINFLUX concept and why it's so fundamentally different from the localization that is typically found in other systems, or Palm Storm and systems Paint, or systems of that kind. Now let's imagine we have a molecule that is in here, and we assume, or we know that the molecule is within this range. This is a very, very legitimate assumption. We know roughly within the diffraction zone, so this region L could be the diffraction length of about 200 nanometers or so. The molecule must be somewhere in between. And so what do? We scan, of course, the donut across this range, this one-dimensional range, to find out where the molecule is located. And so this profile here actually shows the intensity profile of this excitation donut. So don't forget, it's an excitation beam. And of course, once we start moving the donut, of course, then the fluorescence will be, say, proportional to the brightness of the excitation beam. Now, again, so this is the profile in here. Now you will have some fluorescence. It starts out with some fluorescence. And then once the molecule just coincides in space with the donut zero, or I should say once the donut zero coincides in space with the molecule, the fluorescence will be minimal. And therefore, it's very clear where the molecule is located. It is located exactly at the point where fluorescence is minimal. And then we know, yeah, this is the position of the molecule because fluorescence is minimal. So this is XM. Now you may say, okay, but this is kind of tedious. I need to scan very densely across the molecule and have to make the molecule always coincide with the position of the zero. No, that's not the case. It's not a case because we know the profile of the intensity of the donut, because that's pre-installed by the way we make the donut. So this is easily measured, by the way. And since this is known, and in the first approximation, this is a quadratic function here, it's perfectly enough to measure the fluorescence at the endpoints. So we need to measure, say, the fluorescence when the donut is here, donut zero is here, and then we can right jump to the other side of our assumption range where we place the donut now, and then here in this orange point, and measure again the fluorescence. So we need to measure only the endpoints of fluorescence. And then since we know this is a certain function, in the first approximation, a quadratic function, we know exactly where the 0 must be located, because this is a quadratic function, and we can calculate the position xm out of these, say, two fluorescence values, n0 and n1. And so if this range L is smaller than, say, the diffraction range, which is usually the case, smaller than the wavelengths, and if we can assume the quadratic approximation, then we can easily calculate that this position xm scales with the distance L over 1 plus the square root of the ratio of the two endpoints. So this can be easily calculated with a, say, 7th grader equation inversion, and you get this position xm. Now, this is a very interesting equation, although it's very, very simple, because it tells a lot about a concept. First of all, there's no dependence on the wavelength. You see, xm, of course, doesn't have the wavelengths in it anymore. So the parameters that determine xm will not depend on the wavelengths. And because of that, in the first approximation, of course, there is no scaling with the wavelengths. And the interesting thing is it scales, of course, with this range L, in which we assume the molecule to be located. And this is not surprising, because this is where we start out from. And of course, it also scales with the square root, of course, of the number of photons that we detect at the two endpoints in this particular case. Now, if you look and make the analysis of the uncertainty or of the precision that we get, you will see that it scales, of course, with L linearly and with the square root of the total number of photons that we detect, and is the sum of n0 and n1. And so the insight here is very, very profound. It means that the precision that we get scales linearly with the, say, with the range in which we can assume that the molecule is located. And this is something that can be improved continuously. This is very important. You can improve on that, because once you have a certain, say, estimate where the molecule is located after you make the first measurement, you can add a second measurement and bring, of course, the donut 0 closer to the molecule according to this first assumption, or first estimation. And so it's a very, very effective way to increase the precision by just zooming in on the molecule. So the other types of localization, the normal localization, of course, just waits for more photons to show up. So again, we have a scaling here with the square root of the total number of detected photons. That's okay. But, and that's the insight, it's more effective to zoom in on the molecule, to go closer with the 0 to the molecule, than for wait for more photons. In a normal localization, you don't have the option to zoom in, so to speak. But here you have the option of bringing your donut 0 closer to the molecule. And so if you do that, if you have a first approximation, you zoom in, of course, you make the L smaller and the precision goes up. So with the same number of photons, you get a much higher precision. And if you do that repeatedly, of course, you can decrease the number of detected photons that you need in order to get down to the highest possible precision, say, in the 1 to 2 or 3 nanometer range, a region that was not really accessible with standard techniques like Palm Storm and STAT. Okay, now how do you do it in 2D? In 2D, of course, it's not enough to take 2 measurements. You have to have at least 3. So what we've done in the experiments, we have taken 4 measurements usually. They all are indicated here with different colors. So here, measurement here, here, and here. And what we do is actually we measure the detected fluorescence, the fluorescence that is detected here at the detector. And then we can calculate actually where the molecule is located based on the brightness that we measure from each of the 4 coordinates. And then again, we get a first assumption where it is, and the same, say, behavior applies. Now the equation is not that simple anymore. It's a bit more complicated, but the relationship still holds. The precision scales linearly with the range L. In this case, it's of course the diameter with the range L, and inversely with the square root of the number of photons. And so once we have reached a first estimate where the molecule is, of course, we can zoom in and then locate the molecule more precisely, thereby saving many fluorescence emission or if you want to keep the fluorescence emission, we can increase the precision. Now this is actually shown here in a very, say, formal way by calculating the Kramer-Rao bound, which is kind of indicative of the theoretically possible precision that you can achieve with, say, an unbiased, say, estimator of the position with a certain number of detected photons, fluorescent photons, I should say, and the number of photons is shown here in the abscissa, and then you get a precision here on the side, and normally you get for a camera, say, for even the perfect background, say, of 0 more or less, you get only this curve, and for a more realistic background, you get that curve. But for the MINFLUX case, where you have an L of 50, which is not very small, by the way, just 4 times smaller than the diffraction region, you see you get a much higher precision for the same number of fluorescence detections, and this is shown in here. So with 10 detected photons, you get to about 10 nanometers in precision. This is not just calculated, this is also measured. This is why you see those measurement points. And if you make the comparison with a camera, say the normal way of localizing molecules, you would require, in this case, 290 detected fluorescent photons in order to get the same precision. And of course, since this relationship holds, the precision scales linearly with the range L, but only inversely with the square root of the number of photons. As I said, it's more effective to zoom in on the molecule and to chase the molecule with the 0 of the donut rather than to wait for more fluorescent photons as it is done in normal localization. So just by zooming in, making the L smaller, we can increase the precision further and get, say, a more effective way of localizing molecules. Now, once you realize that and everything is in place, and of course we have developed such a microscope in the last couple of years or few years, then you can use it, of course, either for tracking, because that's the simplest way of taking advantage of this, say, super localization that we have in here. Or, of course, you can do imaging, but don't forget the key ingredient to making, say, nanoscale resolution images is to have an on-off transition of the molecule, because this is the way you make molecules distinct, this is the way you separate. But finding out the position in this case is done by localization using a donut. And so MINFLUX nanoscopy uses on-off switching of single molecules, but the way of localizing molecules is much more effective than it is usually with a camera, because we use a structured beam, in this particular case, in this experiment, a donut beam, in order to localize the molecules. Now I'm going to speak first about the tracking, because that's very easily explained. So how does it work? Basically, if you have a molecule that moves around very quickly, we make, say, in this case, 4 measurements, or you can, 3 usually is enough, but let's say 4 measurements. In here, as the molecule moves along, and then we locate the position of the molecule. This has been done actually in here. So this is a relatively lousy, say, transmission image actually of bacteria, just to show you what the bacterium looked like. But what we did actually is we tracked the individual molecule, and you see it's actually quite nicely tracked in here. So what's nice about this is that you have 8,000 localizations per second, and the movie is actually slowed down about, the movie that you see is slowed down about 50 times. So what you see is a ribosomal subunit, and the label was MEOS2, which is typically used in palm. And so why did we use that? Because that allowed us to, say, single out a molecule, an active molecule from many non-active molecules, and we could follow that. And again, there is no way of doing that with the cameras, because the cameras are slow. You have frame times, of course, and not only that, the concept of localizing with pixelated detectors such as a camera requires many more photons. Now, if you get high precisions with fewer fluorescent photon detections. You can also imagine tracking fast movements. And this is a little, if you will, physicist type of experiment to track the position of a fluorophore that is in this case attached, as it is shown in this cartoon, attached to a piece of DNA. And then the measurement points are indicated again by these different colors. And since here the molecule was rigidly attached to the piece of DNA, it didn't move, and the localization cloud, or the cloud of localizations, actually shown in here. So these are the localizations. You see a sigma of 2.4 nanometers within a measurement time of 400 microseconds. This is definitely not bad. And if you make the experiment slightly different so that the molecule is capable of moving, because it's attached now to two pieces of DNA, and this end here, the bridge here, is kind of moving. So in reality, it doesn't move like this, of course, but it's not so easy to do it in a cartoon. It's more like a stochastic type of movement, not a periodic type of movement. But the point that I'm making here is that you can see this, say, degree of movement, or this degree of freedom of movement in this y direction in this particular case by having localization that are spread along the y-axis. So while they were previously just centered in a single region, here they are spread along the y-axis. And again, the calculation says that you need 7 times fewer photons than with, say, a very good, say, camera recording, and you get a precision of the order of 2.5 nanometer here with this dye that we used here, in this case, ATO647N, within 400 microseconds. And this is also showing the single molecule tracks for the x-axis and then for the y-axis, and the y-axis reflects actually that movement along the y-axis. And based on this data, we can make now a cartoon that tracks the actual movement. So this is not just a cartoon with random motions. This is based on the measurements of the positions here of the molecule. And this is just to show you that you can imagine now seeing very fast movements of molecules, labeled molecules, in a spatial temporal domain that was totally inaccessible before. And I think this is very likely to open up an entirely new research field in biophysics and molecular dynamic studies. But of course, nanoscopy or imaging is something that we are very much interested in. And once you put in the on-off state transition, you can make the distinction of molecules. And once you have located a molecule at a certain position, you turn it off, you turn the next one on. So by implementing this, say, stochastic single on-off transition, as it is done in Palm Storm, for example, then you can do the separation. But again, contrary to Palm Storm, we don't use a camera in here. We have, say, if you will, confocal recorder, recording setting in here, and we use this information to find out where the molecule is. And this is by far more fluorescent photon effective than the camera recording, both in terms of the precision and hence of the resolution that we get, and also in terms of the speed when it comes to looking at individual, say, diffraction limited zones. Now here, this is a cartoon of an arrangement of fluorescent molecules arranged with the help of DNA. So this is why it's called DNA origami, that is labeled. And of course, in order to make the separation between the individual fluorescent molecules, we have to keep all of them off and turn just a single one on. And this is done randomly in space. And Palm Storm does that, of course, but doesn't get a spatial resolution because the way the molecules are localized is rather ineffective in terms of the fluorescent emissions. But if you do it the MINFLUX way, due to the fact that we need fewer fluorescent photons, we get a higher precision because the fluorescent photons are, if you will, more informative because they're only used to find out that little difference between the zero of the donut beam with respect to the position of the molecule. And this is why they are much more useful, much more informative than in Palm Storm. Here's another example. If you have just 6 nanometer distances, again, Palm Storm doesn't resolve it, but MINFLUX clearly resolves the individual molecules. Although we are very close to the actual scale of molecules of proteins. So this is very, very exciting. It means that we have essentially reached the ultimate resolution limit of fluorescence microscopy, which is basically the size of the molecules itself. The fluorescent molecules of the order of a nanometer of 2, and of course you have a linker usually, and in some cases you have a nanobody or I don't know what. And that starts to matter now because the resolution is already at a scale of the linker. And say 10 years back and also 5 years back, this was a luxury problem, but now it's a real problem. You have to think about the fact that fluorescent molecule is the only thing that are seen by the fluorescent microscope. So fluorescent microscope do not image the proteins of interest, they image fluorescent molecules. And once they've done that, they've done their job. So you should not expect too much from a fluorescent microscope. You have to keep in mind that the only thing they can do is to image the fluorescent molecules. And you have to think as a user whether the fluorescent molecules really give you the information that you want to get. And so you have to think about the linker and I think about the rest. But once you do that, of course, the information that you get is truly spectacular. Now, don't forget, this is all done with regular objective lenses with focused visible light. And 20 years back, I'm old enough to remember those days, this was entirely unthinkable. But now we have reached the final, say, resolution limit that is imaginable. Many people ask themselves at the time we introduced this concept, but can you do large-scale recordings like large fields of view? And a way of doing it is, of course, to activate molecules in the larger field of view so that they are further away than the diffraction barrier. And then you can locate them individually, and this is done in here. So this is actually a relatively large field of view, and we have localized nuclear pore complexes. There was certain protein in the nuclear pore complexes was labeled with a switchable fluorescent dye. So in this case it was Alexa Fluor 647. And again, you see the resolution here measured with conservative estimates is better than 3 nanometers in Sigma. So in this case, the sample was by actually Jonas Ries and Jan Ellenberg of EMBL in Heidelberg, who came to us bringing the samples, and it worked on the first day. It was really impressive and a lot of fun to see that working so quickly and so effectively. And not only that, the resolution that you get is truly impressive. So it shows that you basically can locate, say, individual labels in here. The resolution is very high, and if you compare it to STAT, which already gives you a high spatial resolution, this clearly shows that's another, say, factor 5 or even order of magnitude better than we have in STAT. And if you compare it with standard confocal, basically there's no comparison. It's just showing how much things have changed. Now, so this is very, very exciting. And to give you some numbers, of course, the median of localization here was 2.3 nanometers, which is not bad in X and Y. But the good news here is that it also works with fluorescent proteins. So this case was M-maple, which is related to M-eos. And this was recorded in a living cell. So not only you can look at fixed cells, but you can also look at living cells, provided, of course, that you can label with something that is switchable. You need the on-off switching, don't forget it, if you want to do imaging. So you have this dye restriction that you need to have something that is switchable or activatable. And of course, the kinetic comes in. But the big advantage of MINFLUX over the normal, say, palm storm dyes is that you do not require the many emissions, because usually the resolution in palm storm is fundamentally limited by the number of fluorescence emissions that you require in order to locate the molecules. But here, the number of emissions that you need is cut down by factors, and this really makes a difference. And so while we are still at the beginning of say investigating all kind of say switchable or activatable fluorophores, we can say that the range of available fluorophores for MINFLUX is much larger than for palm storm, because this requirement of fluorescence, of high fluorescence detection numbers, is fundamentally relaxed in the MINFLUX case. Another good news is that you can also do 3D imaging, as is shown in here, not only XY, but also XY and Z at the same time. And the median here is 2.6 nanometers in this particular experiment. It's not surprising that it's even slightly better than X and Y. So here you have a kind of three-dimensional rendition of the nuclear pore complex, and of course the donut that we use here is slightly different. So there is intensity above and below the focal plane, and the measurement points are not just 3 or 4 points in X and Y. There's also measurement here and measurement here, so above and beneath the focal plane. But we quickly jump from each point to the next, so that the measurement is, of course, very fast and very effective. Now, this is again showing the kind of three-dimensional side view of the recorded data, and and this is actually a nice movie showing again the large field of view and at the same time the three-dimensionality of the recording. Okay, so these are the nuclear pore complexes. Now, another good news is that it's possible to record two color channels. In principle, you can have more color channels if you have a detector that classifies the fluorophores. So you have fluorophores with subtle changes in emission spectrum. You can use those subtle changes for fluorophore classification. And here this was the case with these two fluorophores, and so we could say distinguish the fluorophores by the relatively small shifts in the emission spectrum. And not only that we can have two color recordings, we can also make three-dimensional two color recordings, which is again good news, and I will show you in a few seconds an application that takes advantage of this two color 3D recording. Now, 3D recording, yes, that's possible. And this is just an example of 3D MINFLUX recording of a certain protein. It's called PSD95 in the synapse. So I think all these synapse studies will strongly benefit actually from this high spatial resolution that we get with 3D MINFLUX, and I'm expecting that 3D MINFLUX will be quite essential in the coming years and play a big role in sorting out all kinds of protein distributions, say at the synapse, but also perhaps in the nucleus, in other regions. The physics is there, and the labeling is also there. The labeling needs improvements for the reasons I've mentioned. You need to get a fluorophore very close to the protein target or molecule target of interest. And there are many efforts going on in my lab in order to optimize the labels for MINFLUX. But since most of you, of course, are interested in applications, I'm going to show you now something that is unpublished. So it has been submitted to the proceedings of the National Academy of Sciences of USA. And what you see here is an application of two-color 3D MINFLUX. Now, this is a collaboration with the group of Stefan Jakobs, and the work was done by Jasmin Pape in my lab and Til Stefan in Stefan Jakobs' lab. And it's about finding out the position or the relative position, the colocalization of proteins that are really essential for the crystal formation in mitochondria. So it's about Micros complex, and there are proteins called MIC60, MIC19, and MIC10. And the question was, how are they related in space? Now, since this Micros complex is only 20 nanometers in diameter, so it's very, very small, it's really not possible to get, say, decisive information from the normal super-resolution methods. You need a high spatial resolution. And this is why we harnessed 3D MINFLUX, two-color MINFLUX. And this 3D two-color MINFLUX has a spatial, say, resolution precision in the order of 3 nanometers in X, Y, and Z. And so it allowed us to take data that after, say, evaluation with a mathematical, say, algorithm to assign individual molecules, allowed us to see that the MIG-19 protein is much closer on average in space to MIG-16. So the 19 and 60 colocalize very strongly, whereas MIG-10 and MIG-60 are say, found at various distances. So this is something that has been seen, that has been found as a result of this high spatial resolution. And so the next slide is basically showing that this kind of this system, this 3D MINFLUX system that we have, is really capable of taking 3D recordings. So in this particular case, it's the ATP synthase that has been labeled, and MIG-60, so one of the proteins that I mentioned. And this is just to show to you that you can take a movie. Again, this is a mitochondrion in a human dermal fibroblast. And you see very nicely that the MIG-16 is located more on the outside because it's close to the crystal junctions. We are more on the outside, and of course the ATP synthase is more at the inner part of the mitochondrion. So with that, I'm coming to the end, or almost to the end, and I would like to take this opportunity to thank the people who have contributed substantially to the development of this technology, and of course to the data that I've shown. So Yvonne Eilers and Klaus Quosch used to be PhD students in my lab. They have left the lab. They are now not in academia anymore. They are in industry. But Francisco Balzarotti, who has been a key postdoc in this MINFLUX development, has now started this independent career at the IMP in Vienna. And now the person who really works under microscope and really cares about, say, the latest developments and applications of this setup is Jasmin Pape, and she worked together, as I mentioned, with Till Stefan of Stefan Jakob's lab to make the findings about the mitochondria that you've just seen. Now, with thanking them, of course, I'm kind of wrapping up the message of my talk. Now, 3D MINFLUX is something that will be very important in the coming years. First of all, because you get a spatial resolution routinely of the order of, say, a few nanometers that you couldn't get routinely with other techniques. And not only routinely, you get it with a very, very, very high efficiency of, say, using fluorescent photons. Then, and for the same reason, you can measure molecular movements, and a simple back-of-the-envelope calculation tells us with, say, a 1 nanometer precision, you can get with, say, within a spatial interval of about 10 microseconds if the range that you look at is very small. And this is also something which I believe will be very, very important, because it will be a unique way of looking into molecular dynamics, and this is in a living cell. And I expect many changes and many discoveries will be made in the coming decade as a result of having a way of seeing molecular movements in a totally unprecedented way. There's no other method that can do that, full stop. And then I see a number of applications, of course, mapping molecules, say in the synapse, vesicle nucleus, or in other areas. And again, I think a very, very important field will be measurement of molecular dynamics, protein interactions, movements, and so on. And once you've realized how it works and the conceptual power of this new way of identifying, localizing individual molecules and molecular positions, then you will realize this is something that is going to stay, and it will be really important. It's not just another way of doing, say, super resolution. It is arguably the most effective way of doing super resolution when it comes to making use of fluorescence. And this is why there's many, many applications that I am not capable of thinking of. This is what I'm putting up here as the latest thought. Think about how MINFLUX will change your own research, and you may get interesting idea that may change the course of your career. Thank you very much.
Wow, Stefan,
Hi.
Thanks a lot. That was a truly amazing talk.
Thank you.
Thank you so much.
Pleasure.
So there were a lot of questions that reached us, and the first question is, how many color channels will be possible?
Yeah, so far we have done 2 color channels, and the way that the channels are, of course, gained is by classifying molecules that are slightly different in the emission spectrum. Now, if you have several molecules that can be classified, and this has been done also in, say palm-storm methods and other methods that operate with single molecules, you can imagine having a much wider range. So it depends very much on the choice of your fluorophores. So I can easily anticipate with a single excitation wavelength something like 3 or even 4 classes of molecules. But of course, the other option you can have, say, 2 or 3 excitation beams, so that excite different molecules with fundamentally different emission wavelengths. Once you have managed to co-localize those excitation beams, then you can have a wider range of molecules, so different molecules. So I think that something like 5 or so is something that if somebody tries to do it and desperately wants it, can be done easily.
Cool. Then could this microscopy be applied to densely labeled samples, or is it more suitable for sparse labeling and single molecule tracking, for example?
Yeah, it's a very good question. Now, the separation, the way the molecules are, of course, separated from the surrounding molecules is the same as it is done in, say, in Palm or in Storm or in any other of these, say, single molecule separating methods. So what you need is, of course, one molecule within the diffraction range, so within the donut range, and of course, that is active, and the rest has to be off. And of course, this contrast will determine, of course, your density, so to speak, of labeling, if you have many, many, many of them. So you need a very good on-off contrast. And I'm very optimistic that this can be sorted out much better with MINFLUX than with Palm-Storm, for a very fundamental reason. Don't forget that in MINFLUX, we need fewer emissions. And because we need fewer emissions, we can optimize the cage that keeps the molecules dark. And you don't have this option really in Palm-Storm because you want to have a very good cage, a very good on-off contrast, but at the same time, many emissions. And this is kind of conflicting, you know. But once you realize that maybe I need just 80 or 100 detected photons in order to get a high spatial resolution, I can optimize the cage. And I think this is why I believe that in the end, MINFLUX will be much better at imaging densely labeled samples, yes, with many molecules.
Nice. Next question. What is the influence of the polarization of molecules and beams?
Yeah, that's, I think, another very, very important feature or strength of MINFLUX that I didn't really stress during the presentation that I've just given. So in Palm-Storm, of course, the orientation of the molecule really matters because the molecule emits like a dipole, and then it may say, if it's tilted a bit, then some of the fluorescence goes kind of off the aperture of the objective lens. I'm trying to show it now in the camera. And this kind of shifts the point spread functions around on the camera, and then you get mislocalizations because of orientational issues. Now, because and this is the case because the Palm-Storm localization, or the normal localization, relies entirely on the emissions. But here in MINFLUX, the vast majority of the localization or of the information that we get in order to localize the molecule relies, of course, on the photons that are used for excitation, because we use the photons for the excitation photons in order to define a position in the sample, which is used as the reference for the actual position of the molecule. And therefore, the orientation matters much less. In first approximation, it doesn't matter at all, because it doesn't really matter if the molecule is like this or like that, if it's localized with a 0, you know. And so we have a big advantage here as well. So conceptually, in the MINFLUX case, many say problems that are prevalent in Palm-Storm are sorted out, and this is why I think it's a very powerful concept that is, as I said, is going to stay and will be developed even further and further and further, and will be, of course, something that will be extremely useful and widely used.
Thanks for answering this question. Ben, you're saying that MINFLUX has achieved molecule scale resolution that is 1 to 3 nanometers. What are the next steps in development and what should we expect in the coming years?
Yeah, I mean, of course, this is just an ongoing development, as I mentioned. First of all, what we do now, and we're putting a lot of effort into the lab. It's very clear that you need something, if you want to say measure distribution of protein, you have to be very close to the fluorescent molecule, because as I said, in the end, the fluorescent microscope images nothing but the fluorescent molecules, not the actual protein of interest. So we need something that labels the molecules that we actually want to see, or the user wants to see, very, very effectively. So thinking about effective labeling strategies that are useful for having a very high spatial resolution of a few nanometers, that's one thing. The second thing is, of course, developing dyes or screening dyes and fluorescent proteins that are optimized for the specific conditions of MINFLUX, which are, of course, more relaxed than the conditions for PalmStore, meaning that if you just get 100 or 200 detected photons, that's by far enough, but you need a very good on-off contrast so that you can increase the number of molecules within the field of view and take advantage of this, say, feature of MINFLUX of using very few emissions. So that's the other big development. Of course, speed of recording. This is another very, very important development. You have an advantage here because you need fewer photons, you don't have to wait for long. And then, of course, we address with the donut only places where there is a molecule, so that also increases speed. Yet, so far, at least the things we have published are still on the slow side. So we are working, of course, on increasing the speed of recording, but that will come. There are several ways of doing that, and in the end, that'll work out as well.
Okay, I think to this point, I have two follow-up questions. First, you said it's more on the slow side. What does it mean? Like, how fast is an image acquired?
Well, I mean, so far it's very clear that if you take an image, of course, the number or the recording time scales with the number of fluorophores that you image. The more fluorophores you have, of course, the longer it will take. It's not the scanning per se that takes time, because we have very fast scanners. So we use electro-optic scanning. So the donut, the excitation on it goes from one point to the next in no time. You can forget that. It's very, very fast. But of course, you locate each molecule individually, and that, of course, takes a certain amount of time. So there are several ways of speeding up the process. Of course, optimizing the way the localization is done. So having fewer detections needed in order to get the same precision. There's one way, of course, you can imagine, say, parallelizing the process. So that's another way of doing it. And so there are several ways of tackling that problem. I mean, taking large images. So the one that you have seen that is of the order of, say, 5 by 5 micron is still in the tens of minutes range right now. That's by far too long. But again, if you scale it down and you look at small complexes, you're very fast. And so we work on that and to cut down the recording time, and that'll come down as it has come down for actually all other methods, most other methods. So it's important to realize that for a user, that MINFLUX is a very strong principle. And if the principle is very strong, the technology that surrounds it will be developed to be better and faster and more useful. That's just a course of development, a course of technology. That's more an engineering thing than a scientific thing. But of course, that's very, very, very important for a user. And we, of course, will look into that as well.
Okay, so you're saying that it's mostly now the chemists that have to also work on the dyes to make it.
The chemist is the chemist, of course, but not only the chemist. There is also some things to be done from the imaging side, yeah, and there is something to be pushed, and I think good developers will come up with ways to say get a higher, say, precision per unit time, put it that way. And so I'm very confident that it will work out. Although I should say, right now, if you want to look into a specific, say, protein complex, and look at, say, the distribution of certain proteins, labeled proteins within say a certain range, say the synapse or nuclear pores, whatever, MINFLUX is by far faster than any other method, because you just need a few photons. You go from one step to the next. You scan with the donut in no time, and you come up with a piece of data that is infinitely sharp, basically just at the resolution of the size of the fluorophore.
Okay. Then the last question. Why do you think that MINFLUX localization is fundamentally superior to standard localization by calculating the centroid of the fluorescence point spread function?
Yeah, it's a fundamentally different way of finding the position of a molecule. Before the advent of MINFLUX, people thought, okay, the way to localize a molecule is to measure, so to speak, the blob of fluorescence that's produced on a camera, and they have used, they have just seen localization just as that they couldn't imagine something else. I mean, another way of localizing molecule, and this concept of using a zero that is targeted to a molecule. And using that zero just to localize a molecule is something fundamentally different. As I said, we don't use a camera. We have just a point detector that we use. Of course, that can be expanded to a multi-array detector. That's definitely possible. So I'm not saying you must use a point detector, but the majority of the information that is used to find out the molecule is, so to speak, imprinted by the excitation donut onto the sample space, and use the fluorescence only for refining the position of the molecule with respect to that imprinted space in the sample, in the position in the sample, and this is fundamentally different, and this is extremely effective. So the fluorescent photon, of course, is much more useful. They are more informative, if you will, and this is also why you need much fewer emissions and much less time. And this is something that is not going to go away. So MINFLUX is not just another method of super resolution like many others. It is radically different in the way it works from, say, storm, pond, paint, and the rest of it.
Okay, cool, Stefan. Thanks a lot for all these insights. We got a lot of more questions, but I'm afraid we have to move on now. Thanks again for all your time, for answering all these questions.
Yeah. That has been a pleasure. Okay, all the best.
Thanks to you too. Bye-bye.
Bye-bye.
All right, so now let's move on to our next speaker. As the next speaker, I'm happy to introduce Christian Wurm. Christian is cell biologist by training, and during his scientific career, he was working in the labs of Stefan Hell and Stefan Jacobs on various aspects of super-resolution microscopy and also on mitochondrial biology. Christian is now the head of our applications teams at Abberior and Abberior Instruments. Christian, I'm really looking forward to your talk.
Good afternoon, and welcome to the practical part of today's webinar. In this part, we will focus on MINFLUX imaging and tracking on the abberior instrument's MINFLUX microscope. Before coming to the measurements, I'd like to briefly introduce you into the technique and our product. MINFLUX is a single molecule localization microscopy technique. In contrast to other techniques of this type, in MINFLUX, a donut-shaped excitation beam is scanned through the sample for the excitation of fluorophores. This allows to use about 20 times lower light intensities than those used for other single molecule localization techniques. The main advantage is a high resolution which is achieved in MINFLUX. Now I'd like to come to the MINFLUX development, so to say, from the lab to the product. As you have already heard, the first MINFLUX publication came out in 2017. Then we started the development in 2018, and the product came out in 2019. Now in 2020, we are delivering the first microscopes to customers. To give you an overview of the product, I'd like to show you the video clip we prepared for the product launch. So what is the difference from the research lab to the product? From our perspective, the main difference between a microscope in a research lab and a product is the usability of the system. Following this concept, we have developed a turnkey system which includes the conventional microscope body together with epifluorescence and transmitted light imaging options. Further confocal functionalities are available. To achieve high localization precisions without the interference of drift, we have developed an active 3D sterilization with a precision below 1 nanometer. Further, we have included an ultra-fast beam scanner with a speed above 100 kHz, which is necessary for the MINFLUX imaging and tracking. Last but not least, we have prepared the microscope for many upgrade options like full 3D options and functionalities. On the right, you can see the microscope which we will use for the measurements today. It includes two MINFLUX beam paths at 640 nanometers and 561 nanometers, a MINFLUX scanner and a conventional Galvo scanner. The detection is accomplished through APD-based detection modules. The typical localization precisions that we can achieve with this system is in the range of 2 nanometers or better. With MINFLUX, both imaging and single particle tracking is possible. I'd like to start the practical part with single-color MINFLUX imaging of a cell biological sample. For this, I will switch to the MINFLUX measurement software. As many of you can see, functionalities that are required for MINFLUX have been integrated into our ExpertLine software inspector. In this software, you can see measurement windows in the main part of the interface. They represent the MINFLUX imaging workflow. Settings can be made using the settings dialogs on the right side of the interface. As in many other imaging applications, you start the imaging by creating an overview image. After this, you select the region of interest. Then you mark it with this square and then copy over that region to the confocal close-up window. In this confocal close-up, you check the settings if imaging parameters are right, select the window and start the imaging here. The next step is that you select a part of this image. For instance, here is the single peroxisome. Copy again the region over to the MINFLUX imaging window down here. And then again, you check the parameters which are on the right, shown here, and start the imaging after selecting the right imaging sequence. As you will see now in the first seconds, you will see heavy blinking. And these blinking events are already recognized, shown here in this table. And further, they are displayed here in the upper right image in the first localizations shown here. As you can see, the imaging power or the light power that we are using is very small. Here it's 0.5% of the laser power of the system, which is really minimal. One word to the imaging window on the bottom left, to this blinking window. You can see here frequently bright and dark patterns. I'd like to stress here that we are seeing only a live representation of the blinking pattern. Here it's clearly not a structural representation of the image, but really only a time-wise representation. And you can see thereby that the imaging is going on very rapidly, so many blinking events are recognized here and then plotted on the upper right side in this window. This is as well shown again in this MINFLUX imaging dialog, where frequently a number of localizations are displayed. As you might have seen, I was selecting an imaging sequence labeled indirect immunofluorescence Alexa 647 while setting up the measurement. This imaging sequence already includes most parameters for MINFLUX imaging, including L values, relative MINFLUX laser path, and others. The only parameters you have to select in addition to this is the excitation laser power and potentially the activation light intensity. I'd like to switch back to the PowerPoint presentation and talk briefly about sample preparation and samples now. So the next question would be, what is required in terms of sample for MINFLUX imaging? You will see that this is very much comparable to other single molecule localization techniques. As always, you start with a structure that should be labeled and visualized. Then you are attaching dye molecules to the structure, for instance by immunofluorescence. To do the localization process accurately, it is important to have only one fluorophore in the fluorescent state in the MINFLUX focus during the triangulation process. The final image can be then reconstructed from the positions attained by this localization process. As in other single molecule localization techniques, the trick is to use fluorophores which can cycle between a fluorescent and a non-fluorescent state. But how can you make sure to have only one fluorophore in the on state in the MINFLUX focus? This can be either photoswitchable dyes or proteins which undergo a transition from a dark to a bright state under UV illumination, or photoconvertible dyes proteins which undergo a transition from one to another color. Further, carbocyanines can be used under appropriate conditions. Until now, in MINFLUX, most of the time carbocyanines as Alexa 647 have been used. Those dyes can transition to a dark state by binding a sulfur moiety to the chromophoric system. Under UV illumination, the bond between the fluorophore and the sulfur component breaks and brings back the bright state fluorophore. In the sample we are currently imaging, the peroxisomal protein PMP70 has been labeled by immunofluorescence. As you can see here on the slide, peroxisomes are distributed in the cytoplasm. When you take a closer look to single peroxisomes, you can frequently see elongated organelles with a diameter of around 100 nanometers. When you image them with STAT microscopy, you can discriminate proteins on the membrane and their interior. Unfortunately, it is frequently not possible to discriminate individual protein complexes with STAT microscopy. In the recent time, we have imaged quite a few cell biological samples by MINFLUX. The first samples that we imaged with MINFLUX were subunits of the nuclear pore complex. Those subunits were initially labeled by immunofluorescence with Alexa 647. As you can see here, already with samples created with conventional immunofluorescence, we can gain quite some insight using MINFLUX. But of course, other labeling strategies as the snap tag allows to achieve better resolution as shown here on the right. Other examples are coming from my personal history. Here we image mitochondrial proteins as a protein TOM20. As you can see in these images, individual protein complexes can be visualized using MINFLUX, whereas on the confocal counterpart, only a blurred image is visible. Another frequently used cell biological sample is the cytoskeleton. As you can see here, this also works well with MINFLUX. Further, we successfully imaged neurobiological samples, as here the protein spectrin in cultured neuronal cells. Now I'd like to switch back to the imaging. As you can see, while I was talking about samples, we have acquired quite a few localizations. To shoot out a number, we have already acquired about 14,000 localizations until now. When you compare now the MINFLUX image on the right with its confocal counterpart in the middle, I think the performance of MINFLUX directly becomes clear. In the MINFLUX, individual protein localizations are visible, whereas in the confocal image, only a blurred image of the organelle can be seen. Another thing I'd like to mention to you is a buffer which is used for MINFLUX imaging. So typically, a buffer system which keeps the sample oxygen-free as CLOX buffer is used together with a sulfur component as MEA or cysteamine. As you might have seen in the recent literature, there are meanwhile quite a few of those buffer systems available. As stated before, for me personally, the most important feature of MINFLUX is the superb resolution which is achieved on a daily basis. So I think I'd like to show you the resolution that we have achieved in these images today. For this, I will prepare a line profile here in this MINFLUX image. So I take the image, and you can already see on the right side there is a line profile, and then I select the line profile feature and first zoom a bit into that image. Then I'll select a spot where I want to prepare the line profile as shown here. And as a next step, I will measure here the full width of half maximum here in the center at 50% of the intensity. And if you're reading the line here on the bottom left, you can see I'm measuring 4.76 nanometers of diameter in full width of half maximum. From this value, you can calculate the localization precision, which is typically used for single molecule imaging. Doing this with a measured value, you end up with a localization precision of 2 nanometers in this image. I think this measurement demonstrates the performance and speed of single-color MINFLUX imaging. But of course, for doing cell biological applications and answering real-world questions, dual-color imaging is clearly necessary. And as you know from Stefan's talk, it is definitely possible. For two-color MINFLUX, you typically start with a dye which worked well for single-color imaging. In our case, this is Alexa 647. This dye is then combined with a redshifted dye as CF660C or CF680. For two-color MINFLUX, typically you're using one excitation line and split the detection into two channels. As you can see, the channels are overlapping largely. Due to the fact that you know that there's only one fluorophore per focus, you can easily assign then a color channel to this dye. I'd like to show you the performance of this approach again on some mitochondrial images. In this sample, the DNA was labeled with CF680 and TOM20 was labeled with sulfosi5. So two dyes which have a difference of excitation maxima of roughly 30 nanometers. The results of such an imaging strategy are shown here. On the left side, you can see a confocal image of a mitochondrium. Here already the spectral difference between the two labels is visible. On the right side, you can see the corresponding MINFLUX image. Especially in the close-up, the two dyes and the good resolution of both is visible. On this slide, we have a similar comparison. However, here larger mtDNA nucleotides are visible. Now, I'd like to come to the last part, MINFLUX tracking. As a first example, we decided here to go for tracking of single dye-labeled lipids in artificial lipid bilayers. As you can see here on the left side, the labeled lipid diffuses around and can be tracked. The dynamics of those molecules can be seen in the confocal movie shown here on the right. As you can see, the molecules are moving back and forth very rapidly. Now I will change back to the imaging software and start the experiment. After preparation and mounting of the samples, I will start with a confocal measurement of this sample. For this, I will use the overview functionality again. So first, I select the overview window, then I check the imaging settings, and change the imaging settings where necessary. And then I start the imaging using the record button. As a next step, I select the MINFLUX measurement, check again the settings, then I will select again the right imaging sequence, and pinhole sizes, and then I select the imaging window and start the measurement by pressing the record button again. As you can see, already after a few seconds, the first tracks are acquired. And this basically continues quite a bit. Of course, you can zoom into those tracks and you can download the positional data to work on it later in our software tools. As you can see, the individual track length is clearly not identical for all of those tracks, having very short ones and longer ones. I will keep the imaging running a bit to show you the progress. If you keep it running, you're having quite a few tracks over the time here in this imaging window. I'd like to take a closer look into the data now. If you're doing this, you can see that you are able to image more than 18,000 points within not even 3 seconds of imaging. This means that you are imaging one data point every 100 microseconds. To my knowledge, this is the fastest tracking speed that is currently available in the field. Now, some of you might say that we are still in an artificial environment. For this reason, we decided to try out the tracking in living bacterial cells. And as you can see by these results, this works as well. With this, I'm at the end of this presentation. I'd like to thank you for your attention, and I'm happy to take any kind of questions.
Great, thanks a lot, Christian. This was really an amazing show. I mean, I've seen it several times already, but I'm always amazed about the speed and the resolution you achieve. And sorry I left on my video while you were already talking, but I guess this is what happens if you do such a webinar live. So we got a lot of questions for you. The first question is, or a question we got actually multiple times, is tissue imaging possible with MINFLUX?
Mm-hmm. So obviously that was an early on question, if tissue imaging can be done or not. And I mean, we tried it out quite a few times. And I mean, even Stefan showed it in his talk. So tissue imaging is possible. We believe the reason why it works well is because we are in a confocal system. So we have a confocal pinhole, and therefore we can reject background. I have to admit that we are still pushing towards how deep you can go and so on and so forth. But I mean, at the current point, we're having many application development projects up and running, from testing new fluorophores over new samples from different matters up to then obviously tissue imaging. But yeah, the short answer is clearly it is working, and we are pushing in such a direction as well.
Cool. Next question. In the MINFLUX imaging of the peroxisomes, you have reached a localization precision of about 2 nanometers. Could you please comment on the localization precision in single particle tracking with MINFLUX?
Yeah. Yeah, as you said correctly, so we achieved something like 2 nanometers, which is basically a daily localization that we can achieve on this system for imaging applications. For single particle tracking, it's a bit lower, still very good. So we are here in the range of between 10 and 20 nanometers around for these, yeah, as I said, something like 18,000 points within 3 seconds. So really rapidly for this resolution. And again, that's pretty much standard. So it's nothing like a super experimental system where you need to have hours of alignment before starting the imaging. It's really going to the system, mounting the samples that you've prepared, and starting right away with the imaging, which is really cool. I didn't expect that originally.
That really also brings me to my next question. How stable is the alignment of the MINFLUX setup?
So yeah, as I said, much stabler since originally expected from our side. So it is really that we're checking up the system on a say weekly basis or so. Originally we did it more frequently because we didn't trust the setup when assembling it, so to say. But we found out it's pretty stable. So when checking it up every week, we see we have maybe a minor drift or so, and then we can correct this through the auto alignment functionality of the system. So we're having here quite a few options, but yeah, we're not talking about much, and clearly nothing like aligning 2 hours or so in a row and then starting the imaging, but it's really like push of a button and then go ahead.
Nice. Along these lines, could you please comment on room requirements for MINFLUX?
Yeah, that's an important one. So here in our rooms, we don't have a super stable room or so. So the room is sitting basically, or the microscope is sitting basically in a room where there's quite a bit of traffic. So we're having additional microscopes in that room. People are going back and forth, and we're having basically a standard air conditioning. Air conditioning is clearly a prerequisite which has to be met, because I mean thermal drift is typically the source of drift that leads first of course to focus drift, and then of course to drift issues in the beam passes and so on. So to have a stabilized temperature is clearly important, but we are not talking about a whatsoever clean room environment or laminar flow or whatsoever, but really standard good air conditioning. I believe most imaging facilities could host such a setup without many modifications.
Thanks. Next question. How is drift correction performed?
So I didn't stress it too much. So what we're doing is we're adding fiducials to the sample. So for instance, in these immunolabeled samples or fixed samples, what we're doing is basically we're adding gold nanorods or gold particles to the samples. That's basically the last step before mounting the sample in the imaging buffer. They are adhering to the surface, and then we're having some reflected light path working at around 980 nanometers. So out of the typical imaging wavelengths, and then we can correct through this reflected light path, yeah, to correct the drift basically.
Thanks. Last question. Now, does Inspector software use parallel computation, or is it working only on a single CPU?
Currently, it's working on a single CPU. But I mean, question is more image evaluation, and that's streamlined very much. So here you can really see the online result of the imaging here. Yeah, during the imaging process. So there's no reason to think that you have to wait for hours until you have the image developed or so. It's really coming out, and you directly know is a result coming out or not. If you're having more complicated things to do, maybe later on parallelization is an option, but at the current point it's simply not required.
Okay, cool. Thanks a lot for this great demonstration, Christian, and also for all the questions answered. Of course, we have a lot of questions now that we couldn't answer in the interest of time, but we'll get back to you via email and answer all the questions that we didn't manage now. And of course, if you have any more questions, please don't hesitate to contact us directly. And please also get in touch with us if you would like to demo our MINFLUX microscope with your own samples, for example, either live with us in Göttingen or remotely. So this webinar was part of the Abberior Instruments Summer Symposium. Please don't forget to join us for the next events. And with this, I thank you for your attention. I hope you enjoyed our webinar and see you next time. Have a nice day, stay healthy, and bye-bye.
Bye.